Chapter 7, Section 7, Cell-to-cell signaling. Now, you'll be surprised to know that even prokaryotic cells can communicate to each other. Okay, it's not like we communicate and how we talk and we give visual cues or anything like that. No, what they're going to use are small peptides or some form of organic molecules that will be produced in the cytoplasm and then excreted into their extracellular environment. So it's not a yes, no, are these cells present, but it's going to be more of a what concentration. Low concentrations, the cells of that population will act one way. At higher concentrations above a certain threshold, well, then it turns out the cells are going to alter their cellular metabolisms or maybe alter their gene expression. We see this with biofilm formation. When there's only a few cells on that monolayer that you're starting, the cells act one way. But when they've started building up and they start laying down more of that, peptidoglycan meshwork that we've talked about in previous chapters, well, that's because of increased quorum signals, which is triggering changes in cellular metabolism and an overproduction of certain peptidoglycan compounds. And instead of laying them down just as cell wall, they're laying them down as a meshwork, encapsulating clumps of cells together. We refer to this as quorum sensing. Okay, it's a regulatory mechanism used by both bacteria and archaea and some unicellular eukaryotes. It allows the cells to know, quote unquote know, or as your textbook calls it, assess population density. Low density of cells, low concentration of these quorum signals. High density, high numbers of cells in the population, high concentration of these signals. Why do this? Well, because the different population densities are going to call for different responses by the cells. It's going to call for different or an increasing of the population may call for changes in cell responses or changes in cell functions. How can the cell know what the population is? Well, what's the concentration of these quorum signals? Many of these quorum signals are going to be referred to as autoinducers. The bacteria are going to make these small organic things, these may be small peptides, small organic molecules, and just keep excreting them out, pumping them out. Some of them will come back and then get pumped out again. Low density numbers, well, doesn't change much to the cell because the concentration of these things is low. But as the density gets higher, the concentration due to all of the cells in that population pumping them out means that there's going to be a lot in the environment, and it's going to be more than that cell can counter, pumping out. Now those autoinducers are going to start interacting with proteins and whatnot in the cell itself. So the cell, here you have an Ahl synthase, is going to be pumping out this acyl homoserine lactone, AHL. Okay. Here is our quorum signal. Low levels, low numbers of these bacteria, AH levels in the environment are going to be low. As the numbers get higher and higher, same species, AH levels in the environment are going to get higher and higher to the point that it's going to trigger responses. AHL is going to bind a protein. Protein is going to call for changes in gene expression. This cell will start doing something new, stop doing things that it was doing. So that example, acyl homoserine lactone, AHL. Okay, this is the first to be identified. They found it, they saw that there's others autoinducer, autoinducer 2, also referred to as AI2. You see this one's common in gram-negative species. This one also seems to allow interspecies communication. So bacteria, microbes of different species can trigger responses with each other due to AI2 concentrations in a given environment. Gram-positives and archae have been shown to use short peptides. A few amino acids, dozen amino acids, too small to be a protein, but it's enough to trigger responses. We see great examples of this quorum sensing with Vibrio Fischeri. If I remember correctly, this was a bacteria that was, isolated from fish or around on the surface of fish. It has what's called the Lux operon that codes for a bioluminescence due to luciferase. So this was causing some fish. They found it in coastal waters. If you ever go certain coastal waters at right at dusk, at night, you'll see the waves kind of glow a bit. First, people thought it was catching the moonlight. No, it turns out it's bacteria that are glowing due to this luciferase. And it has to be a high enough concentration of them that they can trigger this response in each other. We see this in Saccharomyces cerevisiae, Candida. Saccharomyces, hopefully you remember from Metrocella Malek. Talking about quorum sensing and whatnot. This is the A factor, alpha factor, the mating signals. Here's the bioluminescence in the Fischeri. It does glow in the dark. Turns out some of the things that will be triggered due to these quorum signals. One great example is virulence factors, specifically Shigatoxin. Shigella toxin, or commonly referred to as Shigatoxin, is a toxin that somehow our friend E. coli picked up along the way. 0157H7, remember the O antigen and LBS? 0157. H7 has to do with another antigen, grouping. Turns out produces AHL-AI3. Wow, AHL-AI3, which is excreted out by E. coli, and when it's in the intestine, the presence of the E. coli, the presence of AI3 and whatnot, cause inflammation. Part of the inflammatory response by the epithelial cells lining the gastrointestinal tract, and you'll see the triggering of production of epinephrine and norepinephrine as part of that inflammatory response. Well, it turns out AI3 and the epinephrine are what are required to trigger Shigatoxin production. Flip side of that, our friend Staphylococcus aureus, staph infection, Staph Aureus. Excrete small peptides, that's it. Quorum sensing molecule. These small peptides damage host cells and whatnot. It's under the control of what is known as auto-inducing peptide, AIP. AIP gets excreted out when it gets high enough concentrations, triggers a response in Staph Aureus. Staph causes Staph Aureus to start producing these small peptides, the expression of the different genes for these small peptides, which go, and that's actually what does the damage to the host cells. So if you look at this, what we see is that here on the left, E. coli. E. coli is not inside the cell. Just the way the artist drew this, it looks like it's inside. No, they're just showing them next to each other. E. coli, under normal conditions, AI3 synthase produces quorum signal AI3, which gets pumped out. Presence of E. coli and some of the other things it's doing while it's there in the gastrointestinal tract triggers an inflammatory response. Part of that's epinephrine, norepinephrine. So the intestinal hormones you produce and AI3 all bind to their sensors. The sensors kick off some kinases. The kinases activate transcription factors, and then you get production of the enzymes that will make the toxin. Toxins then produced in the cytoplasm and pumped out, and now it starts doing damage to the intestinal wall, which increases intestinal hormone levels. Any AI3 there will trigger more and more and more and more, which is kind of cool. The Staph Aureus example. Under normal conditions, what they call basal transcription. So there's this certain level of transcription that is occurring. We see that ARG A, B, C, and D are being produced. Our AIP, our auto-inducing protein, it's pre-AIP, means it hasn't been cleaved and cut up yet. It is produced. It's cleaved and whatnot. ARGB pumps it out. Accumulates in the extracellular whatever environment. Some of it will then bind to ARGC, ARGC, which kicks off a cascade. And then ARGA ultimately gets activated. ARGA then goes and binds and acts as an inducer for even more ARGA, B, C, D expression, ups the expression levels of these beyond the basal level, and then also kicks off the virulence proteins, these proteins which will then go and do damage to the host cell tissues.